Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. So we see each other again through this video to continue on our third chapter, which is cell division. So before this, you have learned about cell cycle. There are two main phases in cell cycle, which are interphase and mitotic phase. Then you have learned about mitosis, where there are four main stages of mitosis, which are prophase, metaphase, anaphase, and telophase. So today we are going to learn about meiosis. So meiosis is quite similar to the mitosis, but there are few main differences. One of them is that mitosis will produce 2 daughter cells, but meiosis can produce up to 4 daughter cells. The second difference is that mitosis will produce daughter cells with a similar genetic content as the parent cells. However, meiosis will produce daughter cells with half genetic content compared to the parent cells. Okay, so at the end of the lesson, you should be able to first define terminologies. So we have 8 terms that you have to understand, which are chromatid, homologous chromosome, synapsis, bivalent, tetrad, chiasma, crossing over, and centromere. The second one is that you should be able to state the stages of meiosis and meiosis 2. Let's look at the overview of the meiosis. You can see meiosis also consists of interphase, where we have G1, where the cell grows. During S phase, the chromosomes are replicated, so you can notice that all chromosome is in form of sister chromatid. Then we'll enter the meiosis 1, where the first division occurs, produces 2 daughter cells, 1 and 2. And then these 2 daughter cells will proceed to the next one, which is meiosis 2, and then each of them will divide again and you will have 4 daughter cells. So let's look at the first term, which is chromatid. So I've mentioned about chromatid before, so let's look at the picture again. Both of these, you can see, this is one chromosome, single chromosome, but in form of chromatid. The second picture is also one chromosome, but it's in form of sister chromatid. So what is chromatid? Chromatid is one of the 2 identical halves of a duplicated chromosome, means half of the structure. But the 2 structures are identical, they're similar. So if there is 2 copies, so we call it as sister chromatid. Each duplicated chromosome consists of 2 sister chromatids. So I've already mentioned this one. Once the chromosome is duplicated, it is always in form of this one. Next term is homologous chromosome. Homologous chromosome is a pair of chromosomes. So this pair of chromosomes are identical in length, position of centromere, and it possesses genes for the same character at the corresponding loci. Why there is pair of chromosome? Because one chromosome inherited from paternal gamete, father, and another one from the maternal gamete. So let's look at the picture. So you can see the length are similar. The position of centromere is also similar. And then they have genes that codes for the same character at the same loci. Loci is the place where genes are located. For example, the red chromosome over here codes for S. So at the same location, the blue chromosome also codes for S. Okay, next we have synapsis. Synapsis is a process where homologous chromosome pair up. So in a non-meiosis condition, homologous chromosomes are not usually side by side. But during meiosis, synapsis will occur. So what happens during synapsis is that the homologous chromosome will pair up. to form what we call as bivalent or tetrad. So you see, the homologous chromosome is side to side. So this is what we call as a process of pairing up. So once it's paired up, it will form a structure named as bivalent. Okay, let's look at this picture. So as you can see, this is quite the same picture, but this actually labels the structure. Synapsis just now was a process of pairing up. A bivalent over here is a structure where homologous chromosome already pairs up. So basically, you have to understand synapsis is a process, bivalent is a structure. Next one is tetrad. Tetrad is quite similar to bivalent, but tetrad refers to the number of chromatid it has. Tetrad is a group of 4 chromatids. Next, we have chiasma. So chiasma is the regions or points at which crossing over occur between non-sister chromatids. So let's look at the picture. So we have homologous chromosome over here, and then once crossing over occurs, you can see this part of chromatid will cross over like this. So where is chiasma? Chiasma is this part where the crossing over actually occurs, where the non-sister chromatid overlap. Then we have crossing over. So this is where crossing over occur. So you can see the overlapping non-sister chromatid. Or by definition, it's actually the exchange of genetic material. So DNA from the red chromosome is passed to the blue chromosome, while DNA from the blue chromosome is passed to the red chromosome. So they exchange genetic materials. Then this occurs between non-sister chromatids. The segments at the chiasma break off and recombine. So the crossing over can lead to 1. Genetic variation and 2. Genetic recombinant. Okay, let's look at the next term, which is centromere. Centromere is actually a structure on the chromosome. So you can see the middle structure over here. So this is centromere. So by definition, it's a region where the chromatid is attached closely to its sister chromatid. So it is made up of repetitive sequences in the chromosomal DNA. So hopefully by now you are clear about all the 8 terms regarding meiosis. So let's look at the process. Meiosis requires 2 nuclear division known as meiosis 1 and meiosis 2. For plant, it will occur in male reproductive structure, which called as enter. Inside enter we have microsporocyte. And for female structure, which is ovary, there's a cell named as megasporocyte. So these 2 cells will undergo meiosis to produce their gametes. In animal, we have ovary for female and testis for male. So meiosis will occur to produce ovum and sperm. As I mentioned before, meiosis will produce 4 daughter cells with half the chromosome number as compared to the parent cell. Because it produces 4 daughter cells, then you will need 2 times cytokinesis. The 4 daughter cells, we call it as haploid, means that it's not in a pair structure. Let's look at the picture. So this diagram over here shows you that the parent cell is initially a diploid with 4 chromosome number. After S phase, so you can see that all 4 chromosomes are duplicated and now they are in the form of sister chromatids. And it begins to pair up. So this is a process named as synapsis. So first, meiosis 1 will occur where you can see it produces 2 daughter cells, and these 2 daughter cells will proceed on meiosis 2. Then they will produce 4 daughter cells in total. Okay, next we have 2 stages of meiosis. So as in mitosis, we also has prophase, metaphase, anaphase and telophase. The difference is that all 4 stages occur twice. So you can see here during meiosis 1, where the main function of meiosis 1 is we want to separate the homologous chromosome, and in order to do that, so you have to undergo prophase 1, metaphase 1, anaphase 1 and telophase 1. Then they split into 2 over here, and these 2 cells will undergo meiosis 2, where at this point sister chromatids will be separated, and they will also undergo prophase 2, metaphase 2, anaphase 2 and telophase 2, and finally after cytokinesis it will produce 4 daughter cells. So you can notice there is a period of continuation between meiosis 1 and meiosis 2. So at this point we call it as interkinesis. There are 5 stages of prophase 1. So we have liptotin, where the chromosomes condensing, so it gets thicker, followed by zygotin, where you can see synapsis begins, where the homologous chromosome is pairing up, followed by packaging, where you can see crossing over has occurred, and then followed by diplotin, where synaptonemal complex, which is here, will break down and dissociate. And lastly we have dikinesis, where you can see the nuclear membrane over here is breaking down. To help you understand further, let's watch the following video.
Many organisms pass their genes to their offspring through sexual reproduction. This begins when 2 gametes unite to form an embryo that is genetically unique from the parent organisms. The embryo then grows into an adult who in turn passes their genetic information on to their own offspring. Gametes are formed through a process called meiosis. The cells that undergo meiosis to produce the gametes are called germline cells. In diploid organisms, germline cells have 2 copies of each chromosome. Germline cells undergo meiosis to produce haploid gametes, which only have 1 copy of each chromosome. These haploid gametes fuse to form a diploid embryo that grows into the adult. Meiosis is just one step in the life cycle of a germline cell. Similar to mitosis, the cells also pass through the interphase, G1, S, and G2 stages before they enter meiosis. The DNA inside a germline cell is duplicated before meiosis begins during the S phase. The duplicated germline chromosomes are called sister chromatids. These chromatids remain attached to each other until the second cell division event in meiosis. There are 2 cell division events during meiosis. The first division, meiosis 1, results in 2 unique daughter cells that have half the amount of DNA as the parent germline cell. The second division, meiosis 2, results in 4 unique haploid cells that only have 1 copy of each chromosome. These haploid cells are the gametes that could go on to produce an offspring through sexual reproduction. Let's look more closely at each of the division events. Meiosis begins with prophase 1. In this stage, the DNA condenses to form chromosomes. Here we see the duplicated sister chromatids joined together at the centromere. They stay fused at the centromere throughout meiosis 1. Next, each pair of homologous chromosomes undergoes synapsis to form a complex involving 2 pairs of sister chromatids. Chromosomal material is exchanged between the 2 pairs of sister chromatids. This event is called recombination, or more commonly, crossing over. After crossing over, the sister chromatids for each chromosome are no longer identical to one another. This is one of the reasons why no 2 siblings, aside from twins, are genetically identical. There are several more key steps in prophase 1. The nuclear membrane begins to break down. Then, the 2 centrosomes migrate to opposite ends of the cell and microtubules appear. The microtubules then attach to the chromosomes. The next phase of meiosis 1 is called metaphase 1. Here, the synapsed chromosomes align at the equator of the cell. The chromosomes align randomly, which results in different combinations each time meiosis occurs. The next phase is anaphase 1. During this phase, homologous chromosomes separate and migrate to the two poles of the cell. Importantly, the sister chromatids remain attached at their centromeres. The final steps of meiosis 1 are telophase 1 and cytokinesis. Here, the cell divides into two daughter cells. Each of these two cells now undergo meiosis 2. Meiosis 2 is similar to mitosis. The first stage of meiosis 2 is prophase 2. Again, chromosomes condense, the nuclear envelope breaks down, and the spindle apparatus forms. The major difference between prophase 2 and prophase 1 is the fact that the daughter cells have only one copy of each homologous chromosome. So in prophase 2, there is no synapsis of homologous chromosomes or crossing over. In metaphase 2, the chromosomes align at the equator of the cell. Again, the alignment is random. Since the sister chromatids are no longer identical, there will be many different possible ways for these chromosomes to align. In anaphase 2, the sister chromatids are pulled apart as the microtubules shorten. Also, the ends of the cell are pushed farther apart as microtubules elongate. In telophase 2, the nuclear membrane reforms and the cytoplasm is divided into the two haploid daughter cells. This division is called cytokinesis. Since meiosis 2 began with 2 cells, and each of those cells were split into 2 cells, we now have 4 unique haploid cells at the end of meiosis. These cells are gametes. 2 gametes, one from a father and one from a mother, may fuse to produce a diploid embryo. The resulting embryo then grows through many cycles of mitosis.
I hope the video has given you a clearer picture on meiosis. Thank you very much. Have a good day.